Hi, everyone. Good afternoon. It looks like it is 3 o'clock, so we are going to go ahead and get started. We have our session today is on experimental design and data collection, building evidence for medicine with Dr. Maying Kong. Dr. Maying Kong is a professor in the Department of Bioinformatics and Biostatistics, and she is also one of our core directors at the LCTRC of the Research Design Compliance and Data Management Core. So Dr. Kong, welcome. Thank you for joining us this afternoon.
Thank you. Thank you for your introduction. Today's presentation is on behalf of the Research Design Compliance and Data Management Core. So the core has supporting staff, Wanyu and Wenqi. Also, we have 4 faculty members from Department of Bioinformatics and Biostatistics, along with clinical trial units headed by Rachel. For this core, we have a specific 3 aims as Dr. Gaskin presented. This M3, that presentation is provide guidance, resources, the training to enhance the capability of LCTRC research in conducting human subject research. We also, in the late fall, we also wanted to do one session about the M2, facilitate and provide guidance on the use of electronic health records and large data clinical data set. But today we focus on the M3, about the experimental design for conducting human subject research. Here is the outline. I will begin with the introduction to human subject research, then followed by experimental studies, observational study, data from form and elements for analysis, and the protocol builder and the database builder. Definition of human subject research, according to the NIH, it involves living individuals. A human subject is a living individual about whom an investigator, whether a professional or student, conducting research. That's a human subject research, try to obtain information or biospecimens through intervention or interaction with the individual, and uses and study or analyzes the information or biospecimens, or obtain, uses, study, analyze, or generate identifiable private information or identifiable biospecimen. This is a definition for human, a human subject. The research involved human subject is human subject research. Example of human subject research including the following: collecting blood, conducting a survey, change participates, environment, so on and so forth. Here, we are in the clinical translational research. NIH definition for clinical trial, it is defined as a research study in which one or more human subjects are prospectively assigned to one or more intervention, which may include placebo or other control, to evaluate the effects of those interventions on health-related biomedical or behavior outcomes. So decision tree for whether a human subject research is clinical trial or not, we could go through the 4 questions. So if you say all yes for these 4 questions, this study is a clinical trial. First, does the study involve human participants? Research? Yes. Then second question, are participants prospectively assigned to an intervention? And the third is the study designed to evaluate the effect of the intervention and the participants. The last is the effect being evaluated, health-related biomedical or behavior outcome. So all these 4 questions, your answer is yes, this is a clinical trial. For human subject research, there are also other 2 categories here. One is a basic experimental study involve humans. That are studies in human that have the purpose of understanding fundamental aspect of phenomenon without a direct application toward clinical process or product. According to that, effective May 26 this year, the NIH does not consider this batch to be clinical trial because the outcome may be different from the defined biomedical and health-related outcome. Before that, batch is considered part of clinical trial. But it's effective May 26, batch is not considered to be clinical trial anymore. Observational study, the third category, for human subject research is observational study. That involve humans, studies in which data are gathered from or about human subject participants, but the researchers do not perform intervention. Observational study can be conducted with human participants in real time or can be performed retrospectively using previous gathered information. The main subtype of observational study include natural history studies. Researchers observe a cohort of participants over a long period. Also secondary research study. Research uses existing data or biospecimen that were previously collected for different purpose. For example, Medicare data, Medicaid data. These all secondary can be considered secondary research studies if we use this data set to conduct research. For observational study, also the repository studies are considered observational study. This involves only the collection and storage or maintenance of identifiable private information or identifiable biospecimen for secondary research. For the 3 study type, we can look at this table. These all involve human participants. And then for observational study, it may not be prospective assigned intervention, but for clinical trial and batch, it is prospective assigned intervention. Also evaluate the effect of intervention. The difference between clinical trial and batch is the health-related outcome. Clinical trial evaluate health-related outcome, but batch does not. Maybe involve the mechanism study that fall into the category of batch, not clinical trial. For human subject study, that we need to protect human subject study. So in order to do human subject study, we need to develop protocols, get ARB approval, so on and so forth. There is 8 exemptions for human subject study that we still need to submit ARB approval, but it is have an exemption. For us, we mainly involve the category 4, 7, and 8. Exemption 8 is secondary research using identifiable information or biospecimen. Broad consent and limited ARB review are And so this for ARB approval, protocol development and the ARB approval, we have a clinical trial unit. They have expertise who can help, which later I will refer you how to contact them. So for human subject research study type, we can classify it into two big categories. One is experimental and another is observational. For experimental, we can classify it into three categories: clinical trial design, quasi-experiment, crossover, that's experimental designs. And observational, we have a cohort, case control, cross-sectional. These are the major study types for human subject study. First, we talk about the clinical trials. Particular randomized controlled trials. The randomized controlled trials are considered the golden standard to evaluate treatment effect. In that RCT participants are randomly assigned to either experimental group or control group. Quasi-experiment, it is experimental design. It is similar to RCT, but lacks random assignment. Often when randomization is unethical or impractical. The third one is a crossover design. Each participant receive both the treatment and the placebo at a different stage, acting as their own controls. For clinical trial, RCT is a subset of clinical trial. Clinical trial recall that definition is that one or more human subjects are prospectively assigned to one or more interventions. The purpose is to evaluate the effect of those interventions and health-related biomedical and behavior outcomes. The clinical trial usually developed by 4 phases, phase 1 to phase 3. The phase 1 is a first in human subject study. It is to find the highest dose of new treatment that can be given safely without causing severe side effect. Safety is the primary endpoint in phase 1 study. For cancer-related treatment, it is the phase 1 subject is cancer patient. For vaccines, so on other area, the phase 1 study usually is a healthy volunteer. It's a small study. Look at whether the drug is safe in human. So usually 10 to 30 patient for phase 1. Phase 2 is considered dose finding. It is a phase 2 clinical trial is done to see if it works to certain type of disease, choosing efficacy as the primary endpoint. Meanwhile, monitor the safety profile of the patient. It is find the dose level for phase 3 study. The subject is a target population for what is the target population the drug is developed for. That's the recruitment. We need limited to that target population. Phase 3 is a confirmatory study. It compare phase 3 clinical trial compare the safety and the effectiveness of the new treatment against the current standard treatment. Side effects of the treatment are also compared. The question is, we wanted to answer is this intervention is better than the standard treatment. These usually is a large study, at least several hundred. If we look at, we can look at the paradigm like the 4 phases. Phase 4 is post-marketing for safety and efficacy. For drug FDA approval, we go through this phase 1 to phase 3. For institute like us, university research initiated research usually is phase 1, phase 2. Phase 3 is a big, larger study, usually is industry-sponsored research. And then our institute, like Brown Council, does participate a lot of phase 3 study, also phase 1, phase 2. But for research initiated clinical trial usually involve phase 1 and phase 2. So I will spend a little bit of time on this phase 1, phase 2 study design. So phase 1 is mainly for dose finding to find the maximum tolerated dose, the safety, the primary endpoint is safety. So the traditional design is the 3+3 design. You have the think about the dose levels you like to try from the lower to high. You start with 3 patients. If the 3 patients no toxicity, then this dose is safe. You move up to next dose level. And if among 3 of that, among 3, one, at least one develop toxicity, then this dose we add 3 more patients. And then if it is 1 out of 5, we only have 1 patient have a toxicity. Again, this dose is safe, move up to next dose level. So over this, develop the 3+3 design with the idea here, just at each dose level, we at most assign 2 cohort of 3 patients, at most 6 patients. And if out of these 6, we have more than 1 patient develop toxicity, this dose is not safe. We need to de-escalate that dose level. If the dose is, if we have among 3 patient, if no toxicity, we say this dose is safe, go to next high dose. If we 3 patients, 1 have a toxicity, we add 3 more. Out of the 6, more than 2 develop toxicity, this dose is not safe. We need to de-escalate the dose. Eventually reach out, we find the maximum tolerated dose. The issue for this 3+3 design is only for 3 or 6 patients. Actually, that do not define maximum tolerated dose. This is a very active area for this phase 1 design. There is new technology to do the clinical phase 1 clinical trial. One methodology we used is the Bayesian optimal interval design called the Bayesian design, that we can we look at if after each patient we calculate the probability of toxicity. If it is we have a target toxicity, also develop lower limit and upper limit. If it is below the lower limit, lambda 1, that's dose is safe. We escalate the next patient to assign high dose level. If the patient assigned at the current dose, the subject the toxicity level is greater than lambda 2, it means too toxic, so we need to de-escalate. Next patient will be assigned to lower dose level. And also, it does not limit to cohort 3 or 6. So, for example, here, in this target population, target toxicity, say, is 0.2. We have developed this lower, the algorithm has lower limit, upper limit, and then we can eventually find the maximum tolerated dose for different number of patients at each dose level. So that's more advanced than 3+3 design. In recent my collaboration with Brain Cancer Center, I recommend use the Boeing design. So for Boeing design, for phase 1 design, the target is to find the maximum tolerated dose, but to find the optimal treatment dose level, it's not maximum tolerated dose. For example, here is the profile like a toxicity. Suppose toxicity is the lower curve, that Y is the probability of toxicity or efficacy, that it's 0 to 1. We put it into the same scale. The target is find the dose which potentially have a similar treatment effect as MTD, but has the lower dose, lower toxicity. For example, in this diagram, it is like the dashed line. This indicates the optimal dose. The treatment effect, look at the up curve, is almost like MTD, but the dose level is much lower than MTD. That's more safe for patient use. So what is recommended dose for phase 3? That is, we come to phase 2 trial. Phase 2 trial, the aim is to examine whether a drug has the desired efficacy to warrant further development for phase 3. The response often binary endpoint for success. Sample size moderate, typically 40 to 100. Phase 2A, initial efficacy assessment, single arm, proof of concept study to see whether there is a treatment anti-disease activity that warrants further investigation. Phase 2B is a randomized multi-arm study to identify the most promising treatment for phase 3. That's have lower toxicity and higher the treatment effect more close to MTD. The 2022 FDA initiated this project Optimus, is find the dose, reform the dose optimization and the dose selection paradigm in oncology drug development, try to find the optimal biological dose. What is the optimal biological dose? Like, for example, in the targeted therapy, the MTD through phase 1, we may find this one, the red one. But if we look at the efficacy, the dose does not need that high. This one has the similar efficacy as MTD, but the dose level is much lower. That is called the optimal biological dose. So the paradigm is moved. Instead of find MTD, we want to find optimal biological dose that will be recommended for phase 3 study. For example, in this case, if the toxicity and the efficacy profile look like this, the MTD, say the toxicity is no more than 30%. From the toxicity profile, phase 1, we may find dose 3 is MTD, but look at the efficacy profile, the dose 2 and dose 3, the effect is almost similar. So we prefer dose 2 instead of dose 3. So phase 2 use find the optimal dose, which is for phase 3 trials. Phase 3 is the purpose is for evaluate efficacy and monitor adverse reaction of new drug and device. In oncology study, overall survival is the gold standard primary endpoint. Sample size usually large. Phase 3 study provide most of the safety data because these studies are larger and longer in duration. The results are more likely to show long-term or real side effect. Phase 3 design randomized, double-blinded usually, with a higher power, 90%, and one-sided type 1 error is 2.5%. Phase 2 usually we do 80% of power, but phase 3 usually we power with 90%. And then approximately 25 to 30% of drug move to the next phase. As mentioned, phase 4, it is after the drug has been approved by FDA. It is an observational study. It is a post-market safety monitoring. Sample size usually several thousand volunteers who has the disease condition. And it is a post-market study. Drug may be recalled due to toxicity or efficacy. So here we go through the clinical trial to compare this treatment effect. It is usually we say that randomized controlled trial is the golden standard to evaluate interventional efficacy. There is core features for this randomized controlled trial. It is one is like a phase 1, we do not involve randomization. Phase 2, later phase 2B and phase 3, we compare with the standard placebo or standard treatment, which do involve the randomized controlled trials. The core feature here, randomization, participants are randomly allocated to either the experiment or control group to ensure groups are comparable and balance known and unknown confounders. So it is in controlled environment. Comparison is made against a control placebo group to measure the true effectiveness of the treatment. So it is randomized to the treatment or control. There is all this known unknown confounder is balanced. So that is the beauty of the randomization. Second feature is blinding. Participant researchers and sometimes data analysts are unaware of group assignment, which prevents bias in treating or assessing outcomes. Also is prospective design. The study allows participants forward in time to observe outcome. And the allocation concealment, the process of hiding the allocation sequence from researchers, preventing them from influence who is assigned to which group, so that avoid evaluation bias. Also, the defined endpoint, the measurable clinical relevant outcome are established in advance. Ethical consideration require informed consent and maintains participants' safety throughout the trial. That's For randomization method, our group can help. There is a simple randomization. It's like you're tossing a coin, but that can result unbalanced confoundings. So so we usually at least we use a block the randomization. Say we randomize block. Within each block, we randomize the patient into different sequence of treatment. Another one common used is stratified randomization to ensure balance and important baseline factor, create a strata and set up separate randomization schedule within each stratum. For example, if we want to prevent an imbalance on age in a study, first we can create the strata based on age, then randomize within each strata separately. Blocking should also be used within each stratum. If we have a more prognostic factor identified, we may use the minimization method. There is deterministic or stochastic method that assign the next patient to the group that minimize the overall imbalance across different variables. The advantage here ensure balance even in small to moderate sample sizes and handle multiple complex or continuous covariate. This covariate adaptive randomization are preferred when several important prognostic factors need to be balanced, providing more convincing and statistically efficient result compared to simple randomization. For the blinding there is the goal is avoid potential bias caused by conscious or subconscious factors. Single blind, it is patient is blinded for the receive treatment or control. Double blind is a patient and the assessing investigator are blinded. Triple blind, including the committee monitor response variable, is also blinded. One example we in our university we participate is this NIH-funded project. We use the covariate adaptive randomization method to randomly assign subject to intervention group and standard treatment group. Variable to consider to balance in randomization has disordered and age. In the statistical analysis, these randomized stratified variable need to be considered through adjustment or stratification to control for potential confounding effect. For example, in this trial so far, we did. If we do not stratify, we did not see the benefit for the intervention. Then we stratify the important factor. We do see that treatment has a better outcome than the standard treatment. Although it's written out, it's a small number of patients, it did not reach statistically significant, but the trend is there. This is about the clinical controlled clinical study. Then we come to the quasi-experimental study design. Quasi-experimental research design like this randomized controlled trial, but the only thing is lack random assignment. You have a treatment intervention group, also has a treatment group, but there is lacking random assignment. Quasi-experimental design identify a comparison group that is as similar as possible to the treatment group in terms of baseline characteristics. So it's not randomized, but we at the end we still like to see whether the treatment has effect or not. How we handle with that, we try to find similar group patient with a similar characteristic as those in the intervention group. So we look at the compare the intervention group versus treatment group, but this control group is not randomly assigned. So there are different techniques to create a valid comparison group, such as regression. Oh, sorry. There are different techniques to create the comparison group, such as this regression discontinuity design method or propensity score matching method. The analysis scale is similar to RCT, but here the control group is we kind of use some statistical approach, find a matched control group that is comparable with the treatment group in all other aspects except the treatment. So here, the fundamental principle in this quasi-experimental design is the control group, the comparison group must be alike in all important aspects, only differ in the treatment group assignment. In practical term, that means the patient in the treatment group and the control group is comparable other than the treatment they received. One is treated, one is in control group. So we have used the propensity score matched method to design quasi-experiment for several studies. What I try to find an illustration to illustrate what is the match. Is like we have the treatment group in the left panel, and then we have a big pool of control patient. We look at their basic characteristics. For each subject in the treatment group, find the control which is similar to the treated subject. Eventually, we find a subgroup which is a group of patient from control group which is similar to the treatment group. Then we compare these two groups. So this is the quasi-experimental design. And the third experimental design study is a crossover design. Subjects are randomly assigned a sequence, or sequence 1 and 2 here. And sequence 1 is first receive treatment, then has a washout period, then receive treatment B. For sequence 2 is receive treatment B, washout period, and then receive treatment A. This uses the patient as her own control. Often a washout period, time between treatment period, is used to avoid a carryover effect. Can have a crossover design with more than 2 periods. Advantage of the treatment comparison is only subject to within subject variability, not between subject variability. Disadvantage is strict assumption about the carryover effect, inappropriate for certain acute disease, where condition may be cured during the first period, drop out before second period. It is appropriate for conditions that are expected to return to the baseline level at the beginning of the second period. Examples include treatment of chronic pain, comparison of hearing aids for hearing loss, mouthwash, treatment for conjunctivitis. This for experimental design. I would like to end up this session with the power and the sample size justification. We often ask to calculate the sample size for experiment. It is we often say the sample size depends on the power, and then also it depends on the alternative hypothesis. We have new hypothesis, we have an alternative hypothesis, like the distribution, and the new hypothesis is the blue one, the red one, and the alternative hypothesis. The power actually is a probability. If the alternative red one, the mean is mu1, we reject the new hypothesis H0. That's the power. It is the probability. So from that, we can calculate the sample size depends on the power, that is 1 minus beta, but often depends on the treatment difference. Say, under standard treatment, it is the response, say, is mu0, and intervention group is mu1. Then another thing is what is the variation of the measurement. So we recommend collect some preliminary data or from literature, can figure have some information on these values, so that the sample size calculation is more meaningful. Similar for phase 2 for binary endpoint. You say what is the response rate and standard treatment? What is the response and rate and intervention group? So if we can get some this value estimation from preliminary data or from the literature, that will be very helpful for us to carry out the power sample size calculation. Then I move to the observational study. Observational study from the statistical and epidemiological perspective, we have a cohort study, case control study, and a cross-sectional study. Cohort study could be prospective or retrospective, follow a group over time to see how exposure affect outcome. Case control study, it is retrospective, compare people with condition cases to those without condition to find the association, to find the common exposure and the disease. Cross-sectional study is snapshot in time, analyze data from population at one specific point. So NIH definition of observational study, as mentioned, natural history study, second research study, and repository study. For this, there is a connection between this study. Let's take a look at the cohort study. Cohort study, we say it could be prospective or also the retrospective. It's if we talk about the prospective, follow over time, compare outcome instance, then for this cohort study, we can also think about, we have a look at the patient who are treated, and then we can find the matched control, so compare the treatment effect. So from this, we can also generate the, using the idea of quasi-experiment design idea to use the data collected and cohort study to do some causal inference. For the case control study, it is you'll find the cases and the control. You'll look back to look at their exposure, usually look at the association between the exposure and disease outcome. Okay, so for this is some, I'm thinking about the NIH defined observational study, how we could embedding this experimental design into this, say, natural history study or secondary research study. From this study, we are able to, using the idea like a quasi-experiment construct, like I do some comparison, like one area we targeted to use is using electronic health records data. We can look at the patient, look at the treatment. Patient receive certain treatment, we can construct certain control, compare the treatment effect. So we can still use some idea like a quasi-experiment or experimental idea to use the observational study to do some causal inference. However, that evidence is not as strong as the RCT, randomized controlled trials. So this is the observational study. Then the after we designed the experimental experiment, we really need a collaboration with a clinical trial unit, like develop protocols for ARB approval, also develop a database, and then record these patient treatment outcome, so on and so forth. But for us to analyze data, what data form we have, I'm thinking about how to address this. We usually collect demographic information, exposure, outcome, confounding variables, and then we can think about each patient. We have these data structure, data metrics. You have each patient, you have a exposure, outcome, all these confounding variables. Then we feed into using different models to look at whether the treatment has impact for the outcome with adjust important prognostic factors. So for analysis, we really need this is the data metrics, patient level data metrics. For longitudinal, most study we follow up over time. For longitudinal data, we can construct, it doesn't matter, but we have the long form and wide form for us to analyze. Then long form, you can think about for patient who have a multiple visit, we record one row for each visit. Eventually, we can feed into to do the statistic analysis. Wide form, it is a patient, you can put the variable at visit time 1, time 2 into different variable. We can easily handle with that. And also the in terms of data integration with omics data, genomics data, the data structure, if you can construct as a matrix form, say you have different genes, and then you have for each patient at each time point, the measurement for the expression level, so on for genes. We can easily do the data integration and the subsequence analysis association with the clinical outcome, that type of analysis. I will wrap up with the protocol builder, as I promised, that we have the group clinical trial unit who has a personnel to help develop protocols for ARB approval. That's core protocol components. And also CTU has this database design and build tool, especially use REDCap, so on and so forth, can help to design database and collect data so that we collaborate and then eventually get the data to do the analysis. So I will wrap up with the flowchart and the summary. That's really the for human subject study. It is a collaborative work among you, biostatistician, us, as well as clinical trial unit, the involved protocol builder, data database management, and for us design the experiment and collect clean data to do data analysis, write the report result, and help interpret the data, help the write a research report, as well as this publication, or for prepare the preliminary data study for proposal, so on. So that is really is a collaborative environment. This can be done only through collaborations. So if you like, yeah, we are available to help here. To contact the RDC core, as you can go to the Clinical Translational Research Center, click on RDC core, there is RDC intake form. Also here I presented my, here is my email address. Also Rachel mentioned, if you want to contact the CTU, here is the email address. RDC core intake form also goes to CTU units. So let's work together to advance clinical translational medicine. Thank you for your listening. I will stop here.
Thanks so much, Dr. Kong. We have a question from Dr. Wong. He would like to know if it would be possible for us to share the slides.
Yes.
Okay, wonderful. If you wouldn't mind sending the presentation to me, I can put it in a PDF format and then send it to everyone.
Okay, no problem.
Are there any other questions before we wrap up? Okay, it does not look like there are. If you do have any questions, feel free. Oh, it looks like we do have a question from Leslie.
Let's see.
Leslie, I think if you unmute, you should be able to ask your question. Oh, never mind.
Sorry about that.
Oh, no worries at all. Okay, so if you do have any questions, please feel free to reach out to Dr. Kong. I'm sure she would be happy to help support you in any way she can. So thank you everyone for attending, and thank you so much, Dr. Kong. It was a wonderful talk. Very much appreciated. Okay, thanks everyone.
Bye-bye.